So today I'll be talking about, that's funny because I'm pretty tall. Today I'll be talking about several different things. One of the big things that we think about with EM is we often think that a lot of these particular techniques, whether it's immunogold labeling, just regular EM, or FibSim or confocal, can't be interchanged or quantified. And a lot of times there's a lot of controversies in the field about like how to quantify things appropriately. So one of the things that we really believe in our lab, one of the big things that we think about in our laboratory is that there always needs to be some type of purpose of why you're doing something, and there needs to be an output of why you're doing it so that we can gain some type of knowledge for this. So when we look at our lab, we have this workflow where we look at TM, and then with this workflow, we develop to do things diagnostically, where we look at human tissue, mouse tissue, and all other types of applications. And then what we do is we try to make a connectome. And so we've been pretty successful at doing a variety of different tissues to be able to do this. We'll debut our first like connectome across all mouse tissues that we've kind of done SBF, SEM on later this year. Obviously, there's a lot of other papers that we've done in between. And then we do a lot of different quantification metrics, and my funding to be able to kind of do these particular things is with CZI. That's my big grant that I have. And then also a lot of funding from Bureau's Welcome Fund and various different iterations. And so basically, the whole application of all this is hopefully to be able to understand and predict mitochondrial structural changes that link to pathology in different disease states. And then also to be able to look at a way to refold cristae morphology. Because a lot of times in EM, one, you only see about 25% of the cristae are altered, like when you're looking at a diagnostic image and there's some variation. So if we're able to, lucky enough to pick that up, the goal is to be able to repair those in tissue. So a big thing in the understanding is like, why would we resolve ultrastructure? Why would we need it, even though we have beautiful confocal imaging? One of the things that you can see here is that this is a SIM image that Dr. Marshall has done in the back, who's my research assistant professor. And this is some ER for SIM as well. But the thing here is that you don't see the cristae all the time, even with super resolution regularly, right? And so sometimes you have to do a couple extra different things like the last talk with expansion microscopy, or you get lucky and then sometimes it just kind of has the right combination. Sometimes I just tell everybody, use MitoTracker green. Don't use MitoTracker orange. It seems to work when we're doing cristae. That's not always the case, but it kind of makes people a little bit more happier. So I do it as a joke. But one thing is that with TM, you're able to do this often. And this allows for you to be able to measure different types of organelle contacts. So what you see here is just ER and mitochondria. But if you notice, if you do the ultrastructure image of it, you can be able to look at the mitochondria and ER a lot closer together. You can actually see the proximity changes. With confocal, one of the downsides is that even if you do proximity ligation assay, you're about 40 nanometers and you're seeing kind of associated interactions, but you don't know for sure. And so this is why you need EM-based techniques to be able to do this. And EM-based techniques are all the resin-based techniques. And one of the big things that I love about ultrastructure is that you can see kind of nice particles. I mean, these are not nice particles, these are HIV particles, but I think it's really cool to be able to, visualize and do EM on them, right? And so the reason this is important is because this was a collaborative paper we did with a lab at VUMC, which is the Vanderbilt Medical Center in infectious disease. Senior author is Dr. Sully Wanjala. But members of my lab throughout the paper, whatever imaging techniques, these are the things we did. But one of the things I just want to show you is the power of EM here is that not only can you look at different types of ultrastructure, you can also go to even a smaller basis to be able to look at viral particles or nuclear pores, seeing what's coming in and out of there. So this brings a question. I call it room temperature EM. I know it's resin EM, but I just name things a little bit differently for me. What still needs to be done better, right? And one of the things that still needs to be done better is quantification metrics. So if we look at this image, of course, we see lysosomes, we see cristae, and we see the double membrane of mitochondria. And so this is all great, but we don't have any other information. So if we just show representatives like this really doesn't give us information on like what's happening to whatever treatment, gene knockout, pathology we're doing. Because a lot of times when people do cristae, they'll say, okay, we do we see kind of some cristae, a little bit of cristae, and this, and then they quantify it and they say, oh, this is it. And it happens repetitively. But it also depends on like what type of 2D slicing you've done to be able to see what you see. So you need a better way to quantify. And it's the same thing with other types of tissues as well. One of the big things is that with EM, you can actually diagnose quickly what type of structures these are. This is an iPS cardiomyocyte. This is a TM image from this is myotubes actually. But the key thing here is that you wouldn't be able to see that at the confocal level a lot of times. Sometimes you can, where you can say like, okay, there's an HEK cell. You kind of see like a triangle type shape, but not all shapes are similar or indicative of what you're actually going to see. So this begs the question about how do we quantify all these different types of structures like lipids, mitochondria, lysosomes as well. And so one of the things we've done is we've made a toolkit for the laboratory. So in our laboratory, one of the things I believe is just in open publishing. Yes, I have like other nice papers, but I don't really kind of care about that. I like to publish wherever so that people can get access to the information to be able to do that. So one of the things is we've published these 5, and there's a lot of other papers that we'll kind of go over in detail, but there's experimental methods that actually test critical observations. And 3 of these we did with Mayo Clinic as well. So, oh, actually, no, sorry. 4 of these we did with Mayo Clinic. And then after that, I've kind of continued on to do some of those particular things. So a good example is this paper that we did in collaboration with the Mayo Clinic in Iowa, where we came up with a way to actually do kind of the not normal things when you're doing EM. So one of the things that we always think about is kind of making a pellet, making agarose gelatin around the cells, and then you kind of do the slicing. One of the things that we thought that was kind of an additional step is, well, maybe we could actually just fix the cells in the actual 6-well plates or a smaller well plate and then pop them out of the resin. It's a little bit harder to do. But then the question becomes like, do you scrape the cells or do you do this particular fixation for this long? And so one of the things we did was try to figure out the best method to figure out what would happen. And so as you can see at the top, to make it more, appropriate for publication, we added insulin treatment. And one of the things just for you to know, just for concept, is that when you give insulin in cardiomyocytes or myotubes, you have fusion that occurs. So these mitochondria have proteins in them called fusins and fissins. Fission proteins and fusion proteins, and OPA1 and MFN1 and MFN2 join together to actually make these larger mitochondria. Okay, so that's just so the background so that everybody's on the same page. And so we compare these because we know that down here, eventually we'll see differences. So we tried so many different things, and you can see that they're basically horrible. So we'll get down to this particular row. So after about 5 minutes and 10 minutes of fixation, they start to look okay. And then we did the entire EM process. And one of the things that we noticed is that about after 24 hours of fixation, you could also see really beautiful resolution of the cristae. I was very happy about this. I was like, yes, because these are some of the first things that I kind of learned. But then we also said, well, it can't only be cells that we fix. Maybe we should do this for tissue. Obviously, everyone's okay with, saying, when you're telling somebody to process things, okay, do 1 millimeter cube cuts and try as small as you can and then fix them. Don't do any large pieces because, you're going to have some artifacts, and we talk about it, but we don't kind of show it in a lot of different details and different ways. And so one of the things that we did with the Mayo Clinic is that we try to talk about different types of artifacts. Now, in the field, John Yatrishna is the first one, along with Jeff Salisbury, to actually find nanotunnel structures. They were originally called MOAS, M-O-A-S, but it's kind of taken off in the field, and now it's called nanotunnels. Just a history lesson for you all. I think it's always important to kind of point out who does what and how it kind of goes different ways. But these type of structures that you see that have these thin membranes with no cristae inside that are connected to these 2 bulbs, if you will, that are mitochondria, this is a nanotunnel structure. And when you have artifacts, sometimes these appear. And other times, these nanostructures are good for diagnostic reasons, and we'll talk about that. But basically, with CO2 versus isoflurane, you have a structural change in your mitochondria. And then when you do the same thing with PBS and PFA, you have a change in your structure. And of course, this is the classic example when you have a small piece of tissue versus a large piece of tissue, then you're going to have a difference in fixation procedure. But this is all good. So now we've kind of fixed this whole artifact situation for mitochondria and things that are close in proximity to mitochondria in tissues and cells. But it still doesn't get us to the question about how to quantify them. So another thing that we did was we kind of wrote kind of like a manual, if you will. I mean, for me, I'm a young PI. I think like 400 something citations is good. So I mean, y'all are probably more senior, so y'all think thousands of citations. But for me, I was like, yay, they're paying attention. So we've done two of these. One that's about almost 400 citations, and one that's right at 100 and something almost around how to quantify mitochondria in general. So we walk them through image J, we take the image, and then we teach them how to identify all the different types of structures, regardless of tissue type. So the same thing works in all the different tissue types for mouse cells, human tissue. And yes, we also, I think there should be comments in the reviews because they asked us about the point method. We do answer those questions that they're interchangeable in the way that we're doing things, just in case you're all already thinking about that. And then we also do quantifications of different types of Christie measurements so that people have a lot of different things to look at. And so what we did was we actually did a summary of the literature to actually describe what people had done and what we thought that would be the best recommendations. One of the really cool ones was from Yuri Hanowski's papers. He developed a Christie score that we kind of included along with Christie number, Christie surface area. But that was really cool because we took it and we modified it for 3D, and I'll talk about that later. But one of the cool things about the Christie score is it's qualitative, but actually quantitative because it gives really strict guidelines to go across versus other things are kind of like, I see this and I kind of see that. And so it's really important that we have that standardization across the field so that when we're telling investigators to be able to say, okay, these things are this, and they want to quantify and publish it, now they have some ways to be able to do that. Then one of the other things is we said, oh, we should do other types of things. So one of the people that's a big guy in the autophagy space was like, are you going to do like a lysosome paper? You should do something and talk about the other organelles. So we did. And one of the things, this is just in fly EM. So MARF stands for MFN2, MFN1 is homologous to it. So basically, when you knock it out, you get this induction of integrated stress response, which is this ER stress response, and it burns off with the ATF4 pathway into something else. But the main point is that once it's activated, ATF4 can induce these changes in lysosomes. And so you get these large lysosomes and small lysosomes, and the cells are much larger, but in the fly, they're much smaller, but you can see that they're increased, obviously, compared to the control images. And these are from indirect flight muscle. And so we basically teach individuals how to do lysosomes, all the other types of recycling machinery that needs to be discussed. So now that we did that in the lab, that was the first year of the laboratory, second year of the laboratory. We wanted to kind of do phase 2, which is apply all of these to 3D world and also apply these to CLIM-based techniques. So one of the things is that we basically developed an atlas that we're about to publish, but we published each individual paper. And what we did was we decided to do young and old individuals to look at morphological changes to kind of get a better understanding of what might be happening diagnostically about shape changes that could be inferred later on to actually use like principal component analysis, histogram distributions to be able to come up with some way to be able to score mitochondria. The current way to score mitochondria is just kind of like phenotypically, and they call it mitotyping. This is something that Martin Picard kind of came up with in Cell Reports in 2019 and 2021. He did 2 papers, one in the brain and skeletal muscle on this. But basically, we've been starting to do this now that we've kind of answered that first question. Of course, we use the same funding. It's always good to give thank you to the funding. And one of the things about TM is that you don't see the entire image. So when you have a 3-month, 1 year, 2 year as just an example, and then you also have this heart failure image where you see altered Christie, you kind of see that all the mitochondria are very similar. However, as you already kind of know, when you do SDF, SEM, or FibSim, you're actually able to see unique mitochondrial shapes. But one of the things that I think that is overdone is that we only describe mitochondria by volume or sphericity. We don't try to actually assign different characterizations to the actual mitochondria by classification of size and or whether or not they're more complex using like shape factor or form factor. And so what we did was we decided to start doing that. So I'm not the first one to start doing this. There were 7 shapes that were published before with Brian Glancy. He pulled a lot of the literature together. He was missing mega mitochondria, and I collaborate with Brian Glancy a lot, so that's not like shade or anything. His former postdoc persona is on here, who I collaborate with a lot. But the point here is that in these particular types of shapes, one of the things that we are not thinking about is we just talk about the shape. And we only talk about it energetically, maybe in skeletal muscle, but we actually kind of have to think about how these shapes actually behave throughout the entire body, how they actually could be designing diagnostic changes. And one of the things that's really left out in the field is describing how cristae morphology is actually inside of these unique shapes. So I thought this would be another way for us to start thinking about this in the pathology world, is that whether or not when you're looking at things diagnostically based upon a certain shape, does that mean that the cristae architecture is different? And does that mean that there's also changes in energetic capacity, an earlier sign for diagnostic capabilities to be here? And so this was just to kind of talk about this in the context of physiology and metabolic changes, but it's something to think about as we discover what all this information means. So in this particular paper, the other thing that we talked about was actually not only talking about phenotype based upon frequency and distribution in a tissue, but talk about how aging alters these mitochondrial structures in different ways. One of the things that I found unique is that we've published like several papers on skeletal and heart, and we think well, this is kind of like a summary in the lab. This is not outside of the lab. We just kind of talk about like what could be a pathological mitochondria. So we're starting to think about this. So this is not published. This is just thought process. But we have done this for cancer because this one just came out in 2026 in iScience. So I can say that there is so much heterogeneity in cancer, even sometimes when you're given a drug treatment, you really don't know what you're looking at. So it's going to take a little bit more further knowledge about like how mitochondria are distributed in a normal tissue to compare to the heterogeneity of what happens during cancer. And you can check that one out in iScience. And I did that with MD Anderson. But basically, one of the things that I thought was unique when I was writing this in circulation research was actually talking about the shape of mitochondria. A lot of the things that we've actually found is that the heart failure samples, they actually have elongated mitochondria, they're more branched. But usually when you're doing the EM slices, and there's only a specific type, I'm not saying what it is yet because we're in the process of publishing it, but other types, they behave differently. So sometimes classically you all might see fragmented mitochondria, or you may see enlarged mitochondria. So it's just something to pay attention to. So we talked about this in the context of like different types of heart failure may produce different types of mitochondrial shapes. And so even though you're looking at it diagnostically and saying there's a pathological effect, but what does that mean? Does that mean that if these mitochondria are all been in this particular way, it's going to be this type of heart failure or this, right? And so just starting to kind of change the understanding. So now that we're kind of phase one going down to phase two, one of the things I want to talk about is the difference with FibSim and SBF-SEM. So we have the FibSim at Vandy and then the SBF-SEM at Mayo Clinic, and I collaborate with Mayo Clinic to be able to do it. We're in the process of hopefully Thermo working with us to get one at Vandy. But anyway, the point is that if you can see, the grayscaling is so much better in FibSim than SBF-SEM. This is generally an issue that's continuously happening throughout the field because when you're doing automation, it's a lot harder to do it with SBF-SEM than FibSim. I mean, yes, FibSim is amazing. You know, you can check out this where we've talked about FibSim and talked about how you can render, you know, anything basically out of FibSim is beautiful. But there's issues when you think about it in the context of machine learning. This is not a paper we did. This was from the Genelia group. Basically, from Jennifer Lipson-Schwartz and others, they were able to assign 35 different interactions that happened in this particular paper, and they were able to quantify this. And they did this with different types of cell lines like HeLa and Jurkat cells. And the thing is that people still need to do this for a tissue, right? So this is something that you're starting to see a lot of where people are doing mouse tissue, but not so much human tissue. I actually reviewed a paper in a certain journal that was like going to be the first liver like diagnostic tissue. It was really good. But the thing is that this was an N of 1, and it was like 35,000 mitochondria. It was a lot, but this is an N of 1. So we really have a long way to go to really understand what's happening. And remember, the advantage of looking with SBF-SEM, you're going to get more depth, right? So then you can be able to cover more mitochondria and quantify. There's much more limited time, much a higher yield of structure and fine details with FibSim, but there's still some issues. Now, you can't directly apply just yet this automation that works for FibSim to SBF-SEM. So this is why we started to do manual segmentation. So one of the papers that I did with Mayo Clinic was getting everybody at least caught up to be able to do manual segmentation and like a protocol to be able to do that so that they could use a mirror software that's kind of user-friendly-ish compared to like doing Dragonfly or Pure Python and having that plug into the Amira software where maybe people may have less experience. So this is just a workflow of just talking about SPF-SEM in general, but we did do this mirror paper just looking at the manual segmentation and the way to do the quantifications so that people would know how to be able to download that information. So now that everybody's caught up with SPF-SEM, the question becomes the low contrast of SPF-SEM does not allow for current machine learning. What has been done, right? So one of the things I like, Uri Maynard is a friend of mine and a colleague of mine. He published his paper in Nature Methods. I think it's absolutely fabulous, where he's using the PSSR method to be able to develop higher resolution structure changes when they're low resolution. But if you notice for SPF-SEM, it's not as good as what you would see for FIBSIM. So this is a thing that we talk about when we meet on campus, like because we have running projects going on, and also when we're at conferences. And it's a big debate of like, how can you actually improve the SPF-SEM resolution when you're not getting the same type of resolution at resolving some of these FIBSIM-like structures? Well, there are other people in the field that have already started to do this. Kadar and also Lucy have done this kind of separately and together, where they have this whole community-based idea of where people should segment, and then together we can be able to use this as a machine learning tool to be able to do 3D reconstruction of everything. And I've donated all of my slices as well, or ortho slices, whatever you like to call it, to the program. So Kadar should have another version coming out that actually does better with MitoNet 2. I think it's already out, if I'm not mistaken, or at least on bioarchives, that allows for you to do SPF-SEM as well. So this is one answer to this question, but then there's other ways that you could do things with empanada. So we've been developing, trying to work with Uri Maynard's PSSR method to be able to come up with a better way of doing resolution and then being able to look at the boundary more clearly. It's not perfect right now. This is something that we're trying to do. We at least know that we can differentially actually look at this pretty decently with SPF-SEM, but we're refining this. Obviously, you can see that not all of it is perfect. So we're hoping to be able to do that and then hopefully get these nice structures like this in the FIBSIM and have the automation go as well. One of the things I also wanted to talk about is the histogram distributions, right? And the principal component analysis that I was talking about. So we're hoping in the future, as you saw those shapes that were assigned, we want to be able to bend them. Now, remember, that's not a finite amount of shapes, right? There could be infinitely more. So let's say there's one mega mitochondria, but it may have a tail attached to it, or it may have a smaller like nanotunnel structure attached to it. So we need to be able to bend them all together around certain types of classifications. And so if you can think about it kind of like RNA-seq or site-seq, you can do a UMAP where you kind of cluster them together. And so that's what we're in the process of trying to do right now with the lab in Toronto, is to be able to kind of cluster them in regions so that we can kind of assign the same type of function. And then, of course, we're using empanada to be able to do this. And then getting advice from Kadar is really important as well. And then this is just an example of like how it does not work so that you don't think everything kind of works in my hands, because it doesn't always. And so this is like one of the initial tests, and you can see it was not good at all. So we've tried, and we've kind of now at least gotten a lot better where it can actually respond directly to SPF-SEM. We're not ready to publish this yet. That's why I said to the end of the year. We're going through actually a lot of different other people's lab systems, in particular like Anna's lab at Berkeley, where she's done fabulous work in the liver space with FIBSIM. We're trying to apply some of her other types of ways to render ER to SPF-SEM and things like that. So we'll debut ours in the fall. But we are able to look at boundary different changes and things like that. So we're actually really comfortable now looking at separation of mitochondria, even if they overlap. So there is some discrimination that's happening with each type of mitochondria, which is great. So now if we apply this back to fixation procedures, one of the things is we want to be able to look at all organelles, not just mitochondria. So in the paper with Mayo Clinic, what we did was we also were able to do fixation on all types of mitochondria and ones that come in contact with other things. So in the blue, this is actin, red is mitochondria. The white spaces are MAMs or MERCs, mitochondria-associated membranes, or mitochondria endoplasmic reticulum contact sites. That's like the hot thing in the field, but there's other organelles that interact as well. Lysosomes and lipid droplets as well. And this green area is mostly ER-SR, depending on how we bend it. But the point here is that when you look at this muscle, and this was in collaboration with this particular part of the paper was in collaboration with Brian Glancy, because him and Christopher Fleck were doing a lot of the FIBSIM. But basically, the key thing here is that we want to think about how we can quantify this in segments. And so we need to take it piece by piece to be able to come up with a database way where people can click on and say, I want to measure cristae or I want to measure lipid droplets. And so that's what we did. So this is still kind of like phase 1.5. So basically, we were able to describe looking at cristae morphology and be able to actually quantify this in the way that the cristae score was. And then we did this in brown adipose tissue where we have to discriminate between lipid droplets and mitochondria. And so the work of this worked pretty well. And we also had other measurements that we were able to quantify this by. We did the same thing for lipid droplets as well with mitochondria in the same brown adipose tissue samples. And then because the editor asked, he was like, well, can you kind of like show that this is linked to kind of the cristae morphology from the previous paper? So we showed that mucose was down. But basically what I'm trying to say here is that there's a lot of different types of interactions that may occur in a mitochondria, may they be primary or secondary interactions with lipid droplets. And so we have to think about this. So hopefully in the future, we'll be able to take different types of tissue samples and use either Mayo Clinic's SBF-SEM or the FibSim at Vanderbilt and be able to come up with a way to create a workflow that allows us to use Python and MATLAB to be able to quantify this outside of a mirror so that everyone can have access to this. And then we have enough samples to get started. Like we have every particular tissue that you can imagine. It's just that we haven't done everything. And we've been pretty successful at doing this. We have heart, skeletal muscle, kidney, liver. It just goes on. Brown adipose tissue. We've done it in plenty of different things. And right now, for the diagnostic piece, because I know y'all are like, how does this connect to what we're doing? So we started to do this in human muscle as well. And so we've done this in the aging cell paper where we classify different types of muscle that come directly from the vas laterus, and we're moving to other types as well. And then we're studying sarcopenia, so we have 2 papers on that, and then we'll move to something else. And this one's linked to actually MFN2. And we've been able to show that there's unique mitochondrial shapes based upon age. And then we have a paper with Brian looking at fiber formation that occurs based upon the structural change that may happen as it sets in with age. And this is just a quick application so that you all can see that how we're applying these samples does matter. So peripheral artery disease, this is a paper that we're about to submit to a good journal where we've kind of picked up on a way to actually interrogate new information about PAD. Previously, it was thought that it was only just an oxygen and vascular disease, but now we kind of know that there's some mitochondrial layers to the disease. So PAD is a way that you can think about systemic vascular disorder where it affects metabolic function and flow, and then it causes chronic ischemia. And then there's mitochondria do have a role in this dysfunction. So the way that we know that it has a role in this dysfunction is we've stained for collagen showing the capillaries are a lot larger in the PAD samples compared to controls. We also found individual staining for mitochondria with COX that were unique and not just kind of scattered throughout in the control. And we saw reduced staining with myoglobin as well. So this is just to kind of orient you about mitochondria. So just remember that mitochondria are dynamic. And so when we're capturing a snapshot, it's not the entire picture, but it's just something just to think about. So we had developed a hypothesis around, remember that mitochondria go through fusion and fission. So we thought that the fission mechanism was a little broke in the overall tissue because we saw that there's some places there were large mitochondria, some places we saw that they were smaller. And so what we realized is that in the PAD-treated samples, we found that there was a distribution that was a lot larger in volume, which would suggest that these PAD samples are going through hyperfusion and sometimes they're not. It depends on the sample and the severity of the disease. And then we were able to see, and these are both elderly patients that are one's normal and ones that has PAD. There's accumulation of lipid droplets that are occurring. And then what we've done, and this is a work in progress that we're about to publish, but we haven't done the rendering completely, but we can see that there's lipid droplets that are accumulating that are consistent with what we see in the FibSim. And mechanistically downstream in the paper, we basically also have knocked out ATF4 because we see that there's this activated ER stress changes, and we see that there's these structural changes that are occurring. So we're about to wrap this whole paper up where we've actually done the entire block. But the point here is that when you have peripheral artery disease, there's a reduced staining of mitochondrial proteins, in particular DRP1, that are associated with an increased severity of PAD. So this is just a snapshot. I'm happy to talk after about this. I want to thank some of my collaborators, Josh Beckman and Alex Sullivan and Craig Brooks, UT Southwestern, VMC, have been collaborators in the context of collecting the clinical trial samples and doing that. Dale and Hanada, they've helped UCLA and Iowa, and Mark at Oregon State. They've kind of helped with different parts. Mark's helped with some of the making sure our processing is good on our coding. Hanada provided one mouse model, and Dale provided another for this paper. And then the first author of the paper is Dr. Marshall, who's in the back. She runs both of my labs, one at Meharry and one at Vanderbilt. And then these are some of the main collaborators from the lab that do different things in the clinical trials. And then I want to thank everybody that's in the lab, and the undergrads, and then the alumni of the lab. And then these are the different years of my laboratory. I'm very young, so it's just not that many years yet, but yeah, but I'll take questions, or not questions. Thank you. I'll wait till the end. So.